Hi, hi everyone, how are you doing? I hope all of you are doing great, and it's nice to see or hear from you all. So, today we are going to talk about the microbial eukaryotes, and it will be a bit similar to what we learned yesterday, a bit as long as what we did yesterday. So, the learning objective is as simple. First, we have to identify which eukaryotic cell type are found in bacteria or archaea, or vice versa. And we have to also compare and contrast flagella and cilia. Then we have to identify which eukaryotic cell structure are found in microscopic fungi, protists or helminths, and then we have to also talk about how to differentiate among the terms regarding fungi nutrition, metabolism, and reproduction. And then we will also try to summarize the relationship of mycorrhizas. And then Then we will between we will distinguish algae from protozoa and also other eukaryotic microbes, and we will then go ahead to differentiate among the key terms regarding protozoa nutrition, motility, and other stuff that you can see on this slide. And in 8, we will describe the two phases of the protozoa life cycle and state why a cyst stage may be useful in a protozoa. And then in 9, we will list and briefly describe the three major groups of helminths. 10, we will identify and summarize the stages of a parasite helminth life cycle using key terms. And the last one, we will state the morphology, nutritional needs, and modes of reproduction in helminths. So you remember yesterday we talked of the domain bacteria and domain archaea. Now we are in the domain of eukaryota. So gear up for the fun of it. So this is basically a generalized eukaryotic cell. From most of your past courses, you generally have an idea of what this cell type, what this cell is, and what organelles it contains. So I won't go much deep into it, but this is the few that we'll be handling. Few that we'll be talking about. Basically, we talk about cilia, flagella, cell wall, glycocalyx, cell membrane. I presume we talked about cell wall, glycocalyx, cell membrane from the past class. And with this time around, something that is new, it should be the cytoskeleton and also ribosomes. So first, let's handle cilia. What is cilia? So basically, cilia is mostly found on the outside of the cell membrane of most of some of these microbes, and then some of these eukaryotic cells, and basically their function is for motility. So they are mostly present on protozoa and in some of animal cells, but not in my bacteria or archaea. And they are distributed over the entire surface of the cell, as I've shown earlier on. And they are actually shorter than flagella and helps in the movement of the cell. So flagella function is also for motility or movement, and then it's found in some eukaryotes, but also in some it's found in some eukaryotes and some protozoa and archaea, and it's also in bacteria and also archaea, but there's actually differences in it within it. It's actually long extension and usually 1 to 2 total. That's the ones that you can find on the bacteria and archaea, and it's not as many as the one that we've seen previously from the cilia. And then in eukaryotes, it's actually involved in movement, basically wimping and bending of motions. And this is also driven by ATP, and it's about 10 times thicker than what we've discussed so far. So now let's zoom in into the flagella itself. Um, it's covered by extensions of cytoskeletal membrane. I'm sorry, cytoplasmic membrane, and then it have a basal body, which is triplets of the tube. So basically here basal body hooked into the membrane, cell membrane, and then it have an outside body which is basically a hook and also a filament, as you can see from here. And then it's made up of about 9 plus 2 arrangements of filaments, which we will go more deeper into it. But counterwise, the flagellum of in eukaryote is more nicely round with this basal body, and within it, you can clearly see the cytoplasmic membrane here, and inside it is the central microtubule pairs. So now we've seen what is helping the bacteria move. We are now zooming into the cell wall. So as we've already discussed previously in the old lecture, this particular cell wall or this cell wall is found in fungi and algae and not in all of the eukaryotic microbes. Their function is basically to support and help in the shape, and they have different composition than that of the bacterial cell wall that we discussed earlier on. And in fungi, it's made up of cellulose plus mixed glycans, and in algae, it's made up of cellulose, pectin, or silicon and silicon dioxide. These are just basically the major types that we can find. But in your own discretion or in your own knowledge, what is the bacterial cell wall composed of? You've already discussed that, so you can make cells you can make notes of this and test yourself. So now we also move past the cell wall. Now we are in the cytoplasmic reticulum, cytoplasmic membrane. So basically, we have the this structure is same has same composition and function as the one that we found in the bacteria, and this is typically phospholipid layer which is embedded in protein molecules. The main function is just selective permeability and transport. So as you can see within it, there are embedded channels, and there's this help in the movement, whether in or out of the bacterial cell. But most eukaryotic cells have no cell membrane, so no cell wall. So you have to put that in mind. They're just made up of sterols or stabilized membranes. Next, we will talk of glycocalyx. So it's also called the extracellular matrix for some eukaryotic cell, and is found on most of eukaryotic cell and outside of the cytoplasmic membrane. So here you have the cytoplasm, and then you have the cytoplasmic membrane. And then outside of the cytoplasmic membrane, that's where you have the glycocalyx. The function is for protection and attachment. So it's composed primarily of polysaccharide, also protein, and there is also some proteins and lipids present. And there's a variability in its composition. So, for example, there might be a slime layer, there might be a capsule like, there might be network of fibers all around. But in our guy, it's more varied. So you have to put that in mind. So now we'll be talking of the ribosomes, which is also a subcellular organelle that we have to discuss. So ribosomes are present and in all cell, and all of them have these ribosomes. For example, in bacteria, they have 70S. In eukaryote, they have 80S. In archive, they have 70S, but they are structurally different. So now let's talk about what the 70S and all these kind of different type of ribosomes look like. So here in prokaryotes, we have the 70S, which is made up of both the 50S and the 30S. So basically, the 30S fit right under the 50S. And then in the eukaryote, we have the 80S, and it's the same kind of conformation. You have basically a top and a bottom. So here 60S, the bottom is 40S. And within each of these 40S, you can find like a lot of subpopulated proteins under it, which I wouldn't go much deep into it because it will be more confusing. But basically, the function is for translation or protein synthesis. So under these, we have the Spinberg unit, which is a non-metric unit for sedimentation rate, and it's actually a measure of a time or defined as 10 raised upon minus 13 seconds. So in biology, the sedimentation rate or the sedimentation coefficient will require to the rate at which a molecular particle travels to the bottom of a test tube under the centrifugal force of an ultra high speed centrifuge. So we use this unit to kind of quantify how much of a work rate these ribosomes have been performing. So they are distributed throughout the whole of eukaryotic cell. They are scattered freely in the cytoplasm, and they are attached into the rough endoplasmic reticulum and inside the mitochondria and also on chloroplast. So clear example of how it looks like. Here within the cell, this is the whole cell. Within it, we have the ER. So now this is the magnification of the ER, and around the ER, we have the ribosomes on it. Now we move to the cytoskeleton. So there are three main types of cytoskeleton. We have microtubules, we have intermediate filament, and we have actin filament. And clearly you can see microtubules is up here. We have intermediate filament and we have the actin filament, and they are all right within the cytoplasmic membrane. And their function is just as simple: anchoring of organs, anchoring of organelles, as you can see here, and they help in the movement of cargo entirely throughout the whole cell, and basically also helps in shaping of the cell and enabling the cells in changing its shape. This is mostly more required during movement. So now, in relation to movement, let's talk more about the actin tails. So this is basically in listeria within the host cells. Listeria within each host cell causes host cell actin polymerization at one end of the bacteria cell. So basically here at one end of the bacteria cell, there is just polymerization of actin, and basically it's helping this bacteria just in order to just propel into the adjacent cell. So now we have one cell here, and within this cell we have the listeria in there, and then actin is the actin basically is activated just to help the listeria to hijack the next cell. So actin is here, it just pushes or propel the listeria species bacteria just to move into the next cell, and basically help in infecting the next cell. Here they are so actually on this in these links, there's actually more videos that you can watch. If it was in class, I would have played it, but because most of you will get tired listening to me talk for over an hour, I will just leave this part for you to always click on the link and watch it. So here is a practice test. You can actually work on it at home, and it's the activity is also available through the Google Doc, or you can just print it in yourself and just look through it. You already have a paper version of this, so you can work through it. So now let's talk about the microbial eukaryote. Under this, we have fungi, we have protozoa, protists, which consist of protozoa and algae, and then we have the helminths. So we'll go through this group by group. You shouldn't worry about it now. But the first we'll tackle is fungi. So I'm sure most of you have eaten mushrooms before, so you have an idea of what a fungi is. But basically, there are about 5 million species of fungi in two groups. We have the macroscopic fungi, which obviously mushrooms fall under. We have puffballs, we have gill fungi, which is being showed here. And then we have the microscopic fungi, which are molds and yeast. So with yeast, their size ranges around 3 to 4 micrometers, and with hyphae, we have about 2 to 10 micrometers, but are connected and can be up to about it can be up to about millimeters or centimeters range in length. So all these eukaryotic cells have flagella, cilia, and then chloroplast. Oh no, I'm wrong. They all have same structures as eukaryotic cells, except flagella, cilia, and chloroplast. So now, how do fungi eat and how do they survive? Basically, they are heterotrophs. So basically, they acquire nutrients from a wide range of organic substrate. We have a subgroup which is called saprobes. They feed on dead organic matters, or basically they are decomposers. And we have parasites which require a host in order to live, and obviously they are going to feed on the microbe on the host itself. An example is the athletic feet on an athlete's feet is being infected with Trichophytum rubrum, and basically the bacteria is just uh, I know it's a bit graphic, but basically the bacteria has infected the host and is living off it. So when it comes to the fungi, basically from morphology angle, there are two forms. We have yeast and we have hemphi. So on the hemphi, they just form the filamentous or fungi. They look filamentous fungi or molds. As you can clearly see, yeast is being seen over here. It looks more like fluffy in nature compared to these ones who look more like who looks more like actin filament or more like a filament or rod-like shape. And then we have the third category, which is dimorphic. Basically, they take forms of either the fluffy nature or the filamentous nature, depending on the environment they've been exposed to and the danger they kind of face. And it's very common in pathogenic fungi. So now let's look at some examples of fungi, the diseases they cause, and also basically how they kind of change. So this picture you see over here is the histoplasma capsulatum. So basically this is a dimorphic fungus. And then just focusing on this image alone, you can see that at 25 degrees, it forms this hyphae phenotypic character, basically like filaments. And then when it's cultured in 37 degrees, it changes back to the fluffy nature or the yeast nature. This bacteria or this fungi actually causes it causes mycosis, basically a histoplasmosis, which results in disease we all know as the Darling disease. And this is actually a lung disease, and which can be actually be severe and chronic and resulting in pneumonia sometimes. And they're actually more this fungi is actually more present in the soil. The mycelium is more present in the soil, and the yeast lives inside macrophages when you get infected. So now let's talk about yeast. So yeast are round to oval in shape. They are about 3 to 4 micrometers in size. They are asexual reproductive. Basically, they kind of duplicate into itself, or basically like cell mitosis. They are pseudo hemphi. Basically, instead of forming either hemphi, they kind of grow or attaches to each other, forming long chains. So as you can see in this picture, they just grow and attaches to each other. And here in C is an image of how their reproduction occurs. So you have one yeast duplicates its DNA, produces a small bud, and then the bud also produces another bud, and then it keeps going. They keep dividing over and over and over, and they keep forming more. Then now let's talk about the hemphi or hyphae. Let's talk about hyphae. So these are long trend-like cells found in filamentous fungi or molds. They are about 2 to 10 micrometers, but connected. But when you connect both of them, they can be up to millimeters or centimeters in length. Some of them are some of them divide by septa, basically crossing of walls, and that can be segmented. Yeah, so back to what I was saying. So some are divided by septum, basically across septa cross walls that segment the hyphae into individual cells, and then they can produce through either asexual or sexual reproduction. As you can see from here, this is a septum which is basically connecting them all together. So you have one organism, one hyphae connecting to the other through the septa. And then here, this is a prime example of a non-septic hyphae, just straight all the way, not connected together. And an example of the septic hyphae is penicillin. And by now we already know what penicillin does. The reason why they have these structures in place is just to provide a balance between compartment and communication within the hyphae. That's why they have the septa being present. So, under hyphae and also under molds, we also have the mycelium, basically an intertwining mass of hyphae that make up the body or colony of molds. So, for example, here is like a lot of molds, and within it you have just a single spore. Then the single spore just move forward, forms a hyphae, and then it keeps growing, turn it into a whole mycelium itself. So the tips of hyphae grow rapidly in direction of food source, basically vegetative hyphae, and then they have a high surface to volume ratio and it aids them in nutritional absorption. Enzymes repel other hyphae and then it helps spread their food source. They just produce enzymes that will repel other hyphae and just happen to spread their food source. An example of this is the rhizopus. And then basically, I want us to first look at the life cycle before I come back to talk more about them. So basically, the rhizospus have this life cycle which involves vegetative stage, basically it being in its grand stage, and then it's adult or mature phase, and then it moves into the reproductive phase. Basically, it produces spores and then you just shed off these spores. So for example, this is your bread, and then you have the mold on it, and the mold shed its spores, and then the spores settle on other part of the bread, and then the spores then germinate and produces more hyphae, and then the hyphae now grows into a more surface hyphae, and it also produces more spores. So basically, that's the whole reproductive cycle of the mold that you see on your bread. But back to here, this hyphae is basically vegetative and it just reproduced through two main modes. So it can be asexually reproduced or sexually reproduced. And then after reproduction, it's also going to germinate and produce more of an adult one. So now let's talk more of the asexual spore formation. So we are two categories for this. We have sporogenesis, basically the formation of spores, and then we have the conidia, which is like the non-formation of spores. So now in sporogenesis, it's mostly it's housed within a sporogium, basically like a pocket. This is a sporogium like a pocket, and within the pocket there's more of individual spores in there. And this whole sporogium is mostly carried by winds or by water, and then it helps in its spreading. It's actually resistant to drying and other extreme conditions, so it helps the whole fungi survive in it a long time of its whole lifetime or a long time of extreme conditions. And now we move to the second category, which is conidia. Basically, these are free spores and they are not enclosed, as we can see from here. And they are actually in many type of form, and basically they are released by pinching off or sedimentation. So this can easily be blew off by air and just help in the distribution of it. So now we finish with the asexual. Now it's time for us to talk about the sexual spore formation. So most fungi have sexual spore formation and they are produced through meiosis or meiosis. The purpose is just that important variations occur when fungi of different genetic makeup combine, and they will end up producing a more of a genetic material which is more beneficial or advantageous to what they previously have. And they mostly do this in order to survive extreme weather conditions or just to adapt to it. Their variation actually, this happens also depending on the variation in methods. So now below here is an image of basically what I'm talking about. Here you have a mycelium here, and then you have a neighboring mycelium, and then these two cells begin to fuse. They fuse and just produce more of a hybrid or a newer one, because now both myceliums are exchanging genetic material through meiosis. They produce a newer breed which can help basically resist or withstand what is being observed. Oh, sorry, which can help it resist or withstand the extreme weather condition or to help it adapt to whatever is already being shown. And then here we are going to talk about the impact of fungi. So most fungi actually have both positive and negative impact, but the negative side is that most of these species affect like our grains and cereals, and they just affect them and let them rot on the field. Example here is a fungi infestation in apple and also in corn. But for the positive side, we need them to make our beers and our wines and to produce gas. They produce gases that causes bread to rise. One example is yeast. We use them a lot in production of bread. And they also we use also fungi to help us produce antibiotics and also organic acid and vitamins. They help decompose organic matter, returning essentially nutrients back to the soil. And then they also form symbiotic relationship with plant roots that increase water and nutrient absorption. So in relation to symbiotic relationship of fungi, an example of this relationship is being seen in mycorrhizas. Basically, you have a fungi and then it's been attached to the plant roots. So what happened is that there is a high this attachment of this fungi to plant root increases high surface area because the fungi is producing more of hyphae and it increases the plant's rate. It helps the plant in the absorption of water and nutrients. The fungi then supplies the plant with nitrogen, phosphorus from breakdown of organic material in the soil. Then in in reply, the plant will now supply the fungi with organic compounds for it to be able to survive. One example of this is being seen in orchids, which have like a stage dependent relationship on mycorrhiza. So this is a practice for you. So please do this. And this is also another practice for you. So we finished with fungi. We are back to protists, and we have to first talk about algae. So algae are unicellular, or you can also find them in they can either be unicellular or multicellular. Their size varies from 3 micrometers to 200 micrometers. They are autotrophs. Basically, they rely on photosynthesis to get their energy. And within them, they rely on the sun through photosynthesis to get their energy. And within them, you can find these cells of chloroplast, which are being present, and it just help them to generate their own nutrients. And they can exhibit all eukaryotic, they can actually exhibit all eukaryotic organelles. Oh, something I really wanted to point out, this algae, which is called Spirogyra, is actually important and it helps in food chain as much as it's also responsible for the production of most of Earth's oxygen. So you can see how important algae are. They are present in diverse marine and freshwater environments, including planktons, and mostly they are non-pathogenic, but not completely. So some human diseases, mainly from marine algae, are being basically caused from their toxins that they produce. An example is red tides. And then, for example, this specific subgroup, which is diatoms, this actually produces toxins to fish. They are parasites of fish and they produce toxins to fish and it kills them. And if a human eats this infected fish, it's actually gonna make them sick. So as much as they have good side, they also have bad side. So now we are going to talk more about protozoa. So these are single cell organisms. Oh, I lost. I lost. Okay, let me share it again. I think I lost connection. Okay. So back to protozoa. These are single cell organisms. They are about 3 micrometers to 300 micrometers in size. And they can have all eukaryotic organelles except cell wall, and majority don't have chloroplast. Some have pseudopores, which is basically an extension of the cytoplasm, which help them in moving. So, for example, here you have this protozoa, and basically they use their pseudopores, helping them basically like a means of having like a leg, and it helps them move. So they actually have organelles that can mimic most of what we already know in eukaryotes, like mouth, digestive system, reproductive tract, and legs, which is like in the form of pseudopores, helping them in moving. So with nutrition wise, they are heterotrophs, and then just a few of them are autotrophs. Most of them are living freely, and they are harmless species in both soil and water. So what they do is they scavenge on dead plants and animal debris, or graze on live cells of bacteria and algae. Some of them are parasitic species, and they live on fluid of hosts such as plasma and digestive juices, and they actively feed on tissues. There are about 6,500 species of protozoa being present. And now we have to now discuss how protozoa is being how protozoa basically reproduce. So they are having two different categories. They have sexual and asexual. So let's tackle first of the asexual. Asexual just basically comes from binary fusion, which is similar to mitosis. Basically, make two one protozoa make copies of themselves and split it into two. And then we have the multiple fusion, where there's multiple mitosis happening in the parent. So we have let's say this is a one protozoa, divides its DNA, divides more, keep dividing till it kind of eventually makes a lot of daughter cells, which are then present and distributed, helping it to grow. Now let's talk more of the sexual reproduction. So sexually, they do this by conjugation, basically exchange of genetic material between two few cells. So let's see these two neighbor protozoans, and they attach to each other, and they just exchange genetic material and produces a new one. So with the life cycle of a protozoa, in its first form, in its first form or initial form is the trophozoite. So then trophozoite keeps feeding, and basically due to maybe let's say dryness or lack of, it keeps eating until there's no nutrient available for it to eat. Then the cell begins to run up and like move less because there's actually no nutrients for it to feed on. And then it starts developing cysts or formation of walls between its cell walls. And once the cell wall within its cell wall or these cysts have actually been formed, it turns into a mature cyst, and then the cyst will break out. It's basically gonna stay like in a cyst shape to withstand all these extreme conditions. Once nutrients and moisture is available, cysts break out of this initial cell wall. It crafts its own way up, breaks up, and then come back to its normal protozoa state, which is now actively eating and then causing infections. However, some groups of protozoa, um, their whole life cycle does not include this cyst stage. So the cyst phase is actually protective in poor environment, and it actually help them, but not all of them have this cyst stage that helps them to survive. Now let's talk about classification of protozoa. The first classification is flagellate. So flagellate basically means their source of locomotion is through flagella or via flagella. And these groups are produced sexually, and they involve their whole reproduction system involved in a cyst stage, and then some of them are pathogenic. Some of these examples include Paranema, and all these examples as listed below. Please click on these links and watch the YouTube video. So I'm always trying to make the lecture short, but have the needed information. And with the rest of the videos and the self-test that you have to do, you can work on it on your own schedule or time. So let's talk about the importance of flagellate to the pathogen. So this pathogen, which is Giardia lamblia. It causes gliadiasis. Basically, you wouldn't even experience fever, but you experience diarrhea, and it's coupled with abnormal abdominal pain. And then lastly, it results in a lot of flatulence. It's common in about 1.2 million. It's more common, and we have about 1.2 million cases in the US per year. Transmission of these protozoa is typically through the cysts. So you end up ingesting the cysts whether through water or food. This is more common with hikers who, go to hike and they see clean water and they assume it's a clean water and consume it. If you are not lucky, you end up consuming a cyst of a protozoan and getting yourself infected. And then we also have this species of protozoan, which is Trypanosoma brucei and Trypanosoma brucei. So their mode of transmission is mostly via vector borne, that is blood sucking insects, which you can see from here. The first one, which is the Trypanosoma brucei, causes sleeping sickness, which is basically trypanosomiasis. And then what happens is you sleep a lot, and then it will eventually lead into neurodegeneration. You have paralysis and end up dying. And then the second one, which is the trypanosoma gruesi, causes the Chaga disease. It's basically it bites you, you get an acute phase which is made up of nausea, fever, and few weeks or months later on you have asymptomatic phase, and then complications and autoimmune diseases. These are just chronic phases that you experience, and then you die. This bug causes that disease, and basically it's called a kissing bug because it most of the time it end up kind of biting your face, and this one causes the sleeping sickness. So now let's move to the second class of protozoa, which is ciliate. So remember the other one had flagella. This one have ciliate, and then it's also basically both flagella and ciliate perform the same function, help in movement. And then this group produces asexually through fission. And then yes, their whole asexual reproduction also involves cysts for most of them, and they are not pathogenic except the Balantidium coli, which is pathogenic. So over here, you can appreciate how diverse they are. Like for example, in Paramecium, you can see a lot of cilia all over its cell wall, and then here the euplots is also have a similar one. Please click on this video and watch it. So now let's appreciate some microbes that fall under this group of pathogens. One is the Balantidium coli, which causes balantidiasis, which is mostly asymptomatic to mild. So people with this disease have prolonged diarrhea, abnormal pain, occasionally a preformed colon, a perforated colon. Transition is through fecal or oral, basically having a food that is contaminated, and it's more frequently infect pigs, and it's more rare with humans, unless you end up eating the cyst yourself from the pig. I'm not saying for that reason don't eat pork. I mean, all pork that come on the shelves are nicely inspected and is clean. So the next category is amoeba, and then the mode of its movement is through pseudopods, basically force feet, as we've discussed earlier on. Their reproduction is through asexual fission, and yes, their reproduction also includes the cyst. Some of them are pathogenic. Please make sure you watch this video to have a clear sense. Have a clear sense of what we are discussing. Yeah. So, for example, you can clearly see this amoeba is moving in this direction with its pseudopods. So let's appreciate some of these amoeba. One is the acanthamoeba, basically the brain-eating amoeba, and basically what it does is it causes mening meningoencephalitis. Basically, brain it eats your brain up, and you end up having seizures. So it's present in free living water, and you actually get it if you go around swimming in every single lake that you see. It's rare, but when you have it, it's 100% mortal. Is you result in death. So once you have it, it's 100% mortality rate. You're gonna die. This basically amoeba forces itself into the nasal passages and utilizes the olfactory nerve to enter your brain, and then the rest goes on. Eats all your brain up, and you end up having disease. So, under the classification of these, we have the sporozoa. Basically, it doesn't move at all. So you realize that what we've discussed so far, they are moving, they are having flagella, they are having cilia, they are moving up and down, they are doing all kinds of stuff. But now we have the last group, which basically doesn't move at all. It doesn't have any way for it to move. Basically, it does this by gliding or twisting by no known locomotion organelle. So it just twists or glide itself around. It have complex life cycles, which can include asexual and sexual stages. And the life cycle also include them having a cyst stage, and all of them are pathogenic. And to appreciate some of them, we have the Toxoplasma gondii. And then is this protozoa is worldwide distributed. It causes toxoplasmosis. It's usually asymptomatic, causes sore throat, nerve node enlargement, and you have a little bit of fever. Its infections, its infection is in fetus, and it causes immunodeficiency, and which is often fatal. There's a 33% chance of mothers transmitting this to the fetus, so this is actually a bad news. And if a mother will have it, it's more present within the first to second trimester, and it's gonna result in a stillbirth, which is basically as a result of liver and spleen enlargement, and the liver fails, and then um, the fetus end up dying. Next one is the Plasmodium falciparum. This is more common in sub-Saharan Africa. This group causes malaria. Basically, they infect or they attack the red blood cells and causes damage. And their symptoms actually occur within 48 to 72 hours of you having the infection. You have persistent fever, cough, weakness for weeks. And then if you are not lucky and you have the cerebral malaria, some small blood vessels in the brain are being obstructed, leading to coma and death. It's transmitted by the female Anopheles mosquito, which basically end up biting you and transmit it to you. And it's restricted to just a few belts around the equator. As you can see, this is the mosquito. So you should practice this on your own. Normally in class, we would have hit the 45 minutes and I've given you guys a 5 minute social media Gen Z break. But since it's an online lecture, you can pause, go 5 minutes and come back and come and continue. And now lastly, we tackle the last group, which is Hemoth. So Hemoth are macroscopic, about 1 millimeter to 25 meters in length. These are multicellular animals which are equipped with organs and organic systems. They have reproductive tracts which are fully developed. They also have this primitive non digestive, excretory, nervous system, and muscular system, same as humans. And they are multicellular, so they are made up of individual cells that can have either flagella, cilia. However, they don't have chloroplasts or cell walls. Mm, you can appreciate that one single organism have all of these internal structures, testes, ovaries, and then like almost everything that humans have. I call them the mini humans because they have a lot of things that we do. But basically, there are three groups of hemos. You have the cystoids, which are tapeworms, we have the trematodes, which are flukes, we have the nematodes, which are roundworms. Their source of nutrition varies because they are heterotrophs. They can eat right. They have a wide range of nutrition. Mostly they are parasitic in nature and they are just free living. And one example is the C. elegans, which is a free living worm. It basically eats a bacteria and it lives in the soil. These are more kind of common tapeworm that we will find in the soil, especially when it rains or something. They usually come up. You can see them. So to talk more about the reproduction, they are sexually reproductive. For example, nematodes are known to have both male and females, and then the sexes have different morphology. So an example of them is here. You have a male, which have testes and the sperm duct, and then the other one is also a female, have ovaries, uterus, and then other reproductive organs needed for reproduction to occur. And then we have nematodes, which are made up of fluids. Their sexes are separate, or you can have them as hermaphrodites. So basically, they have both organs being present. And here to pinpoint it, this is an example of how they will look. They might look like. Um, you can have they having a female part and at the same time a sperm duct. Together, they can either interact with other trematodes and reproduce, or they can do it by themselves. And then we have cystids, which are tapeworm, and they are generally hermatro they are generally hermaphrodite. So same as the other one. But the trematodes are both can be separate or hermaphrodite, but for cystids, they are all hermaphrodites. I'm sorry, I lost the signal again. I don't know why my computer keep doing this. So now let's talk about the life cycle. The life cycle begins from having a fertilized egg to a larva and then to an adult. As simple as that. So you have an adult, it produces an egg, egg turns into a larva, larva then turns into an adult. And their source for human infection include food, water, infectious animal. Beyond, you should know that their definite host is humans, and within humans, that's where most of the time their mating occurs. Their route of infection can be oral or through any broken skin or damaged skin. So their transmission can be through the body of the host, or intermediate, where like the host in which the larva develop is like the secondary host, and then they move to a definite or final host in which the adult grows and then they mate. So, like an example, the life form or the reproductive tract of a tapeworm, you can see over here, we have the tapeworm, which is present currently. It's present here, it's in old nature. Let's say it's in its own nature in a grass or something or on a leaf. Then it's being ingested by either cow or the pig. Inside the pig, it grows, produces the cyst, and the cyst then infect. As you can see from here, the cyst now infects these mini hosts into its tissues and every part of it. Like it's just present all over the tissues of the host, like whether it's a pig or a cow. And then the cyst is present, and then a human eats this meat. Now the cyst then moves into the human and basically goes in there and reproduce, grows as an adult, and then reproduces as an adult and produces more of its eggs. So now you have the eggs again, breast cycle. So from eggs is being eaten by a cow, cyst is being produced inside the tissue of the cow or pig. Pig gets eaten by humans. Inside the humans, cyst grows into an adult tapeworm. Tapeworm reproduces, and the cycle keep going on and on and on. So, an example of this, also a similar trend is seen in the pinworm. where um in its native or fertile egg form, um, it's been ingested by the human, basically scratching of contaminated places. And then within the human, the eggs grow, develop into an adult, reproduce, lay small eggs, and then the human brings the egg out through the backside, and then basically it goes on the grass and kids go and play. Some of them touches their mouth and they end up eating it. And then you infect your neighborhood, like your neighbor kid, or you yourself, you end up eating it back and recycles all over and over. Basically, the pinworm is about 2 to 12 millimeters in length, and it causes entabiasis, which is like uncomplicated intestinal infection. Now, let's understand the distribution of Hemoth. So there are about 50 species of Hemoth parasite which are present, which parasitize humans, and they are often geographically distributed or higher incidence in tropical areas. So with humans, we have had a constant presence of Hemoth, and it's only recently that we had Hemoth-free existence. And they've been known to contribute to autoimmune immunity and allergies. So you can appreciate that eating these helminth, and they are causing a lot of problems in your gut. They actually they share components of immunity. So they share components of immunity. For that reason, when they infect you, basically what they do is they have some part of their system that is designed like the immune cells of us. So basically, they cause the immune cells to basically over respond or inappropriately attack self antigens, which you end up having like your immune cells attacking yourself, because this hemoth comes in with IgE, which look like similar of what we already have, which will then trigger all this whole autoimmunity. So now this raises the question is, are we too clean? This is something you have to ask yourself and come up with an answer. So this one you also have to practice all by yourself. And then this ends today's lecture. Thank you for staying with me. Hope to see you on Friday. Remember to do all your post-lab assignment and your pre-class assignment. See you on Friday.